Hi guys, in this video we'll be looking at exam style questions for the membrane transport practical.
Welcome back guys. So we've covered already the planning stages and the implementation stages for these experiments that measure the effects of cell membrane transport and the factors that affect them. What we need to do now is go over some exam questions that refer to these planning and implementation stages. So we've designed a question here, I'll read it through. So it says, a student wishes to investigate the effects of different sucrose concentrations upon osmosis with pieces of swede, swede being a vegetable. They use a cutting machine to form identical cubes of swede before immersing them in sucrose solution. So part A says, describe the method they can use to investigate the osmosis in different sucrose solutions. So this is 5 marks, and it's very similar to the method that we referred to in our previous videos, but this time we're using swede instead of potato. So in general, these types of plant cell osmosis experiments carry out the same format, and as long as you know the method and the reasons for each step of the method, you should be fine to answer a question like this. So because this is just describing the method, it's just a case of listing the different steps. So I'll show you what I've written here. So firstly I've said, weigh the swede samples or swede cubes initially, and then record the initial weight into a table. So you have to make sure that you do know the weight of each cube before you start the experiment. You then place the swedes into the corresponding sucrose solution, or sucrose concentrations, using tweezers, and then you begin the timer. So that's usually a couple of marks there, starting the timer and placing them straight into the given sucrose concentration. And then after an allocated time, or a set amount of time, you remove the swede cubes and record the final weight, and then you calculate the mass change to determine the direction and the extent of the osmosis. So it's a very summary sort of method there, and it doesn't really go into a lot of the equipment used, but it's just asking you to say how could they use these swede cubes to investigate the osmosis. So don't be too specific, just make sure that you cover each of the 5 steps crucially, and talk about how you might want to throw in a little bit of how you use the equipment, for example using the tweezers, how you use an allocated time, etc, etc. But as long as you cover the main stages of how you can use the masses to find out where the osmosis happened and in what direction, then that should be absolutely fine. So part B says, identify 2 variables which need to be controlled in this experiment. So it's only 2 marks, so you don't need to elaborate, you just need to identify those 2 things that need to be controlled. Whenever you see a question like this, think about what you're trying to investigate. In this case, we're investigating how the surrounding sucrose concentration affects osmosis. We're not looking at temperature or pH or surface area to volume ratio. So when those factors aren't being investigated, they should be the ones that need to be controlled. If they're not controlled and they vary every time, we can't be sure that our results are due to the factor we're trying to look at. It might be due to the factors we haven't controlled. So the 2 variables I've chosen to mention here are the time allocated to each one, because if you give one more time than the other, it may allow more osmosis and more change in the mass, and the temperature as well. So the temperature, as we've said, can affect diffusion, osmosis, active transport, all of the pathways, so they have to be kept the same for every single test tube. So that's 2 variables you could talk about there. Part C then says, in terms of validity of results, give 1 reason why using the cutter is better than cutting the shapes by hand. So validity, remember, is one of those words that you have to understand. There's different words: validity, reliability, accuracy. Validity basically means if something's valid, it means that we can take it seriously. It means that the procedure and everything that we tried to follow in the instructions was followed appropriately and correctly. If the experiment was carried out weakly, it wasn't really followed to the letter, and it was carried out poorly, then we can't really say that any of our results and conclusions are valid. Accuracy refers to how correct they are to the true value, and reliability refers to often repeats and replicates and how reliable they are based on how often that trend or that result crops up. So how can we make the results more valid? So why does using the cutter rather than cutting the cubes by hand make the results more valid? Well, when we use the cutter, it makes sure that the swedes are identical in shape. So that's the first mark I've said. With the cutter, it makes the swedes the same shape, so they all have the same surface area to volume ratios because they're exactly the same dimensions. And if you cut by hand, it's less likely to ensure this. By cutting by hand, you can probably get some vague cube shapes, but the surface area to volume ratio won't be identical. So the reason that this makes it more valid is because the surface area to volume ratios can affect osmosis. As we've said before, they can affect diffusion, osmosis, and active transport. So now we've controlled this, we can make sure that our results are more valid because we can say the results are more likely to be due to the sucrose concentrations and not the surface area to volume ratios. So the next part says, suggest why sucrose solutions are used and not glucose solutions. So this is one of those suggest problems. So you may not have come across this thought process in your head before. You may have never have thought about why do we use sucrose and not glucose. But you have to just think about the topic that we're talking about in terms of osmosis and what we're trying to look at here. Why is it better to use sucrose than glucose? So always put it in a bit of a context. Well, what we're trying to measure is the idea that water is moving from where there's more water to where there's less water. And the sucrose solutions obviously are affecting how much that happens. The more concentrated ones will give water from the potato to the solution, and the more watery ones will allow water to move into the potato. So if we use sucrose, there's a reason why we don't use glucose. We're trying to look at how the water moves in between the potato and the solution, but this assumes the fact that sucrose doesn't move because the mass change we want to assume is due to water. By using sucrose, we know the mass change isn't due to sucrose because it's too big to move through the membrane. If we used glucose, we should know that through digestion and different movements across the cell, glucose does actually move through cells, plant and animal cells, through facilitated diffusion. So if we used a glucose solution, glucose and water would be moving between the solution and the potato. So we couldn't use our results to safely say that osmosis occurred and that the mass change was purely due to water. We would not know if it was due to glucose and water or just water. So the way I've written that then is that the membrane may be impermeable to sucrose, which means that any mass change we record is due to water and osmosis, and that's what we're trying to investigate. Glucose can cross the membrane, so this could cause changes in the mass unrelated to osmosis. So the whole purpose of the experiment is to study osmosis and the movement of water. If we use glucose, the glucose may move in high concentration to where it is in low concentration, so that might affect the mass itself. So that would be investigating not only osmosis, but glucose movement as well, and that might be something we'd measure in a different experiment. So it's all about just making sure you know exactly what you're studying, and how, if you use certain chemicals, that might alter what actually changes your results. This is a sort of control. We don't want to alter the temperature in this experiment because we're not looking at how temperature affects the movement, we're looking at how sucrose concentration affects it. So in the same way, we don't want to know how glucose movement would affect the masses, we want to look at purely water. So we have to use something that can't move between the potato and the solution. So sucrose is a better option than glucose. You may not know this off the top of your head, and because this is one of those suggest questions, you're going to have to draw your own conclusion that there must be a reason why he's used sucrose, and that reason must be due to the fact that sucrose must not be allowed to move between the potato and the solution. So part E says here, the student's results are shown in the table. So we've got a table where we've got our sucrose concentration, we've got the initial mass of each Swede, the final mass, and the change in mass, and then we've got a few of the percentage mass changes calculated here. So part E says calculate the remaining values within the table. So the only two values we need to calculate here are the percentage change in masses for the final two sucrose concentrations. So remember, a percentage change in mass is equal to the change that we observe, so the change divided by the original value, times 100. So we'll do the 0.8 moles per decimeters cubed one first. So the change in mass we have for this one is 0.01, and the original value was 1.37. Timesing this by 100 gives us 0.01%. And you know that this is the right number of decimal places because all of the other given percentage changes are to 2 decimal places each time. For the 1.0 moles per decimeters cubed value, we've got the change, which is 0.39, divided by the original, which was 1.33, times 100, and this gives us 29.3%. And you might want to put a minus in front of these each to give you the correct answer, because this is actually a loss in mass. Moving on to part F then, it says the scales measure to an accuracy degree of 0.01 grams. It then says calculate the percentage error of the mass change for the sucrose concentration of 1.0 mole per decimeters cubed. So we need to look back at the table and look at the 1.0 mole per decimeters cubed solution and work out the percentage error of this change. So remember, the percentage error is going to be degree of uncertainty divided by 2 over the measurement that we've made, and then times this by 100. So the uncertainty measure of this piece of equipment is 0.01, so divide that by 2 is 0.005, and then dividing that by the measurement that we made for this one, which was 0.39, and then timesing that by 100 should give us the percentage error, and that value comes out to be 1.28%. So this is quite a small percentage, so the error of this piece of equipment is actually quite small, but that's essentially how you work out the percentage error for a question. So moving on to part G, it says the student knows the water potentials of the sucrose solutions. So remember, in our experiments we talked about, we now compare the water potential of the sucrose solutions to the ones to work out the one of the potato, or in this case the Swede cube. So just look at the table briefly. We've got our sucrose concentration, and as we can guess, the water potential goes down as it gets more concentrated. And again, we've got those units of kilopascals. So he concludes that the water potential of the Swede must be around 0.4 moles per decimeters cubed. Give two ways he could modify the experiment to make the conclusion more reliable. So what you might want to do at this point is just look at how he's come to this conclusion. So he thinks it's about 0.4. So if we look at 0.4, which is minus 950 kilopascals, back up at the original table at 0.4, there's barely any change in mass. So compared to the others, this is probably the most likely water potential of the actual Swede. So two ways that he could modify the experiment, I'll show you now. So one of the points that I've raised is to carry out a test and do several repeats for each of the sucrose concentrations, and then repeat the weighing of each potato before and after. Anytime you can get replicates or repeats in there makes the conclusion more reliable every time. And the second point I've made is to use more sucrose concentration intervals. This would give us more precision about where the actual water potential lies, and gives us a bit more confidence that it would be around the 0.4 mark. So any of those two marks would have been fine. The next question says, which sucrose concentration test is likely to be an outlier result? Suggest two experimental errors that could have led to this anomalous result. So let's look back at the original table, and we need to look at something that fits outside of the trend. So we're going down in our mass change here. It's getting, there's a gain here, a smaller gain, a very, very tiny gain if no change at all, a loss, and then a really small loss, and then a bigger loss. So the outlier is likely to be this one here, which is the 0.8 mole per decimeter cubed sucrose solution. So that answers the first part of the question. So I'll show you what I've written here. So which sucrose solution is likely to be the outlier? I've put 0.8 mole decimeters cubed. That answers the first mark. Suggest two experimental errors. So I've put that perhaps when weighing the potato, we may have said that there wasn't as much loss because some of the sucrose solution may have resided on the cube, so it wasn't shaking off as much as the others. And that was one of the areas we talked about in our video, about how solution can stick to the side of the potato cube, or the Swede cube in this case, and it might add to the weight and incorrectly identify that there wasn't much loss at all. The second point I've put is that the weighing scales weren't calibrated properly. So anything along the lines of the equipment not being calibrated, or double checking it, or even weighing it against zero, or even setting zero before you put the potato on, they're all valid errors that can occur. So that would give you a third mark there as well.
Hey guys, I hope you enjoyed the video. If you are looking for an amazing A-level biology resource, join me today in my series of engaging bite-sized video tutorials. Just click the SnapRevise smiley face, and together let's make A-level biology a walk in the park.